A century ago, science created an animal model to explore the mysteries of the human brain. This model is the laboratory rat. Thanks to the rat, we've made major breakthroughs. In the field of neuroscience, it is today at the center of crucial issues. Can we read our brains? Is it possible to master our fears, our emotions? Will we someday be able to control machines through thought or be controlled remotely ourselves? At the front line of research, rats experience all of these upheavals before we do. What would you think about a machine that could record your dreams and then play them back to you like a film? For rats, this is no longer science fiction. 72 electrodes implanted in its brain record the activity of thousands of neurons. Unlike all other parts of the body, the brain has no receptors for pain. In spite of this invasive equipment, the animal continues to behave like a normal rat. With these electrodes, we can now read its brain, at least to some extent. This dream recorder was conceived by Professor Matt Wilson. Matt Wilson has two passions in life: poker and studying animal dreams and memory. In both cases, you have to imagine and decode what's happening, what's going on in somebody else's brain.
We can tell what the animal is thinking about. What is the brain? What kind of information is the brain processing at the moment? We are eavesdropping on the conversations that are being carried out amongst individual brain cells.
The experiment is deliberately made very simple to isolate specific events in the rat's brain. The rat is placed on a circular track. Electrodes register its neuronal activity while it explores its new environment, particularly when it stops at a specific place for a few drops of chocolate milk. Here, Matt Wilson is looking at a structure in the brain called the hippocampus. The hippocampus is like a huge dispatching center, selecting what to retain out of the millions of bits of information it receives at each instant. Inside the hippocampus, there are place cells, neurons associated with specific places. Using these place cells, the brain creates a kind of virtual map of the rat's environment. When the rat explores the track, a series of neurons fire in a precise pattern that is unique to this place and this animal. Our memory and representation of space are first formed here in the hippocampus.
So that pattern really represents that animal's experience in that location of that particular room. When we see it appear again, which is extremely unlikely to occur by chance alone, we guess that the animal is replaying a memory.
The rat replays these unique patterns during its dream cycles, at the same speed and in the same order.
But there's also this state where the animals are awake, but they're sitting quietly. They're not actively engaged in behavior. They're just they're thinking about what they might do. What we've noticed there is that these patterns are also replayed, but they're replayed in the reverse time order. That is, they think backward. Like, you can think of it like a rapid rewind. Also very compressed, but reversed in time. You want something that's going to look for...
At the moment, it takes Matt Wilson's team weeks to analyze a dream sequence just a few minutes long. But soon they will be able to do it in almost real time.
The idea that sometime in the future we could have software running right now that would tell us exactly what the rat was thinking about, might say dreaming about, is certainly possible. As I say, it would be possible today. In the final analysis, the only way to demonstrate that these memory events during sleep are actually involved in memory that influences behavior is to manipulate them.
After reading a rat's brain, Matt Wilson's next step is to find a way to write in it, to lay down a phantom sequence. An electrical stimulus that replays a play sequence is sent to the rat's brain to see whether it affects the animal's memorization and behavior.
Our work is directed explicitly toward understanding or cracking this code. That is, how does the brain form and use information to guide behavior? That's what we're looking at. We're looking at patterns of brain activity that are related to behavior, that are derived from behavior in the form of experience, then ultimately change behavior in the form of learning and decision-making.
The central quest in the neurosciences is to crack the neuronal code of our conscious and unconscious thoughts, to determine how we learn, memorize, and make decisions. Matt Wilson is only one among hundreds of scientists who are working in this field around the world.
In the end, the question of whether what we learn in the rat ultimately will tell us anything about what goes on in the human brain, I think there we simply have to ask, how different is a rat brain from a human brain? And in the end, what we see is that it isn't that different. The largest difference is that it's smaller, and that there are certain areas that just aren't as developed. But all the basic pieces are there. So what we are studying in the rat ultimately is very likely to be directly relevant to what we see in humans.
To truly explore the inner workings of the brain, you need to use a live model. Just as in poker, you need to ante up. Knowledge always comes at a cost, and the ante here is the rat.
The role of these rats in research is really that they have been invaluable. There's no way that we could do this work if we were not using these animal models.
But why, out of all the possible animal models, did science pick the rat?
Many of these insights were first gained through observation of humans or other organisms. Ultimately, these things were always taken to rats in order to gain the detailed insights. So while humans may have raised the questions, ultimately rats were able to provide the answers.